Hello, my name is Andrii. I'm bachelor student in Taras Shevchenko National University of Kyiv. During this summer, I was working on the project supervised by Dr. S. Aslar from CERN. I would like to thank everyone who made this internship possible for me, and especially my mentor, Dr. S. Aslar, for her guidance, help, patience, and the opportunities she opened up for me. So the idea of our work was to measure the differential cross-section of photon production in association with big quarks in hard proton-proton interaction at 13 TeV, using the data provided by CMS experiment at LHC in run 2. Measuring the cross-section ratio of events with 2 big quarks and 1 big quarks provides an important test of perturbative quantum chromodynamic predictions, in addition to the parton density functions of big quarks and gluons. Having the last cross-section measurement of this process from ATLAS experiment at 8 TeV allows CMS physicists to perform the analysis urgently, but at the same time robustly. The analysis will cover the entire data taken period of run 2 with a total integrated luminosity of 137 femtobar. The challenge of the analysis is the existence of the irreducible quantum chromodynamics multi-jet events as the largest background. So it is crucial to obtain a reliable separation of signal and background events with minimal reliance on Monte Carlo events generators. But the fact that we have very few discriminating variables between useful signal and background makes the task a difficult one. So we need deep learning to solve the problem. The focus of the present project is to find optimal deep learning models to be used for the separation of signal and background events. As we see from the distributions, there are two energy regions of our data. In one of this region, which is in dark blue color on this plot, QCD background is dominating, and in the other one, the signal of photon events we are interested in is added to background. Our strategy is to take background from data in control region and signal from Monte Carlo simulations in signal region to train our deep learning model and then predict QCD events in signal region of real data. Before predicting real data, we checked if our model works properly using events simulated with Monte Carlo. But before deep learning, first of all, we should learn how to read thousands of root files and to read them as less time consuming as possible. To achieve this task, we were using COFIA. COFIA is a Python package based on columnar analysis. The difference of columnar analysis compared to simple looping over events is that corresponding values of every event are transformed in one array, and you can act on this array as you wish, and it would be much faster than acting on every event one by one. COFIA provides also the COFIA processor module, which allows users to worry just about the actual analysis code and not about how to implement efficient parallelization. The model provides a nano events interface and an accumulator utility to reduce the outputs to a single result. In COFIA, we also can set cuts to needed variables which are transformed, as it was said before, in columns. In my work, I was choosing events with one photon more than 1 bjets and H over E more or less than 0.04 for different regions. We can select only needed variables by zipping them in one variable. It will also decrease time of processing. Then we are creating file set with root files. I have more than 1,000 files in some file sets, so I put them in JSON files. But if there are few files, they can be put directly in code. Then we define a processor runner and output to get our data. After taking data goes deep learning. Our preliminary deep learning network, deep neural network model looks like this. There are different input variables, some number of deep layers with some number of neurons per layer. Deep neural network creates connections between input variables and giving us binary output. All deep learning was done in TensorFlow Keras in our work. Input variables, number of deep layers and number of neurons per layer are not the only parameters of deep learning model. Such parameters are called hyperparameters, and we were searching for optimal combination of them to see the best performance of our model using grid search. Grid search isn't something special. It's a loop which is changing parameters of our model and then training it and storing results. In the end, we can see which combination of hyperparameters gives us the best results. Here are listed values which we tested one by one using grid search. But not every hyperparameter was tested alone. Some of them are connected with each other. For example, optimizer, weight initializer, and learning rate are influencing the kernel of the model, so should be tested together. In order to increase precision of the results of our training, we were using k-fold cross-validation. This algorithm divides our data on 5 folds, like on the illustration, but it could be 10 folds or any other number. It's better not to create too much folds because test folder should contain from 10 to 20% of all data. Otherwise, it won't test properly. So during the first step, we are taking model, we are training model on 4 first folds and or on last 4 folds and testing on the first one. Then we are testing on the second one and third one, and so on. In the end, we have 5 results. We are calculating average 5 results of accuracy of our model. We are calculating average of these results and receiving the most accurate assessment of our model's performance. Cross-validation also allows us to use full data for training, so it's a very useful method. I implemented in grid search. Here are the best model parameters obtained from the grid search. We are using all available input variables of photons, jets, and bjets. Adding more input sometimes could affect the model badly, but in our case, it's the best choice. On this plot, we can see real and predicted photon PT distribution from the Monte Carlo signal region comparison. All events here are simulated, and from this plot, we can see that our model's predictions are much like the actual events. So we could move forward to the real data, where predicting accuracy is expected to be even higher. First of all, I need to say that our work is on its final stage, but not finished yet. Right now, I'm working with real data and can present the results of training model with QCD taken from high H over E region of real data and the signal simulated with Monte Carlo. model's ability to distinguish QCD background and signal is very powerful. Total accuracy, as we can see, of our model's prediction is close to 100%. Sensitivity, which shows the ability to distinguish a signal, and specificity, which shows the ability to distinguish QCD background, are also very high. So in conclusion, I want to say that though our project isn't finished yet, we expect to gain high results when we'll predict full real data. We would also see how good does Monte Carlo simulators with real experiments. So this could lead to need and maybe way for improvement for them. Thank you for your attention.
Thank you, Andrei. Any questions for Andrei?
Hey, this is Dino. I would have a short question. Sure. First of all, thanks for the nice talk. On the slide where you showed the graphical depiction of the neural network that you were using, it looked like you were not using MET. At least it was not one of the input nodes. Is that intentional, or is it just the way it's depicted?
No, we were using MET.
Okay.
I just forgot to set it here. Thank you for your improvement.
All right. Any other questions for Andrei? Nice talk, Andrei. So
I can give a brief introduction, maybe.
That'd be great.
Yeah, so I was his mentor. So Ernest is a third-year physics bachelor student from the Taras Shevchenko University in Kiev, and I had the pleasure to work with him on a fellowship program over the last 3 months. And we worked on a project analyzing data from the detector control system of ProtoDUNE, which can be distinguished from the, let's say, primary parts of the detector. And it records information such as temperature, pressure, and high voltage in the detector. And this data is crucial for the reconstruction of the primary data, but it is stored in a database with a very high granularity. So usually there is some kind of algorithm that runs on it, reducing the data volume before it is then fed into the actual reconstruction algorithm. And Ernest focused on the use case of the high voltage system and finding unstable periods of it. And yeah, he will tell you all about it now.
Okay. I think I can start, yeah?
Yeah, of course.
So my name is Ernest Sorokun. I'm a third-year student bachelor of Taras Shevchenko National University, and I'm working on a project named like data reduction for ProtoDUNE detector control system, finding unstable periods in the high voltage system. My mentor was Lynn Gerlach. And to start with, I'm going to tell you a little bit more about the experiment itself. Like DUNE is deep underground continuum experiments, which based on 2 laboratories at USA, like Fermi Lab in Chicago and Stanford Underground Research Facility. The distance between them is around 800 miles one to each other, and they're trying to understand how the neutrino oscillation works. And this is one of the main reasons why these experiments exist. It's going to not exist. Is going to be around at the 2030s, around that year. So the main goals of this experiment is to determine CP violation in the neutrino sector. CP violation is a characteristic of charge and parity symmetry of the particle. Also, one of the goals is to investigate supernovae and search for proton decay. So how these neutrinos are going to be detected? As we know, neutrino is such a small particle to compare with others, and it has such a small mass. it's almost not collision with other atoms. Especially for it, that would be built liquid argon time projection chamber, one of the biggest in it's going to be one of the biggest in the world. How it works? When neutrino goes through this chamber, there is some chance that it could collision with that would be collision with other particle with argon atoms that can produce like ionized particles as well as muons and electrons. And when they're drifting in this, when they're going in this argon, they're ionizing electrons that are drifting by the high voltage electric field to the anode grids. And when this grid absorbs the electrons, there's some signal, a current signal by which we can rebuild the trajectories of these particles. Because we have different angle between these grids, we can rebuild a three-dimensional trajectory of these particles. So for now, there's we have a prototype of this experiment that located at CERN and we name it ProtoDUNE. This is the largest existing prototype of LIRTPC. And one of the points of this prototype that it's located at the surface. So that's why it's littered by the cosmic radiation. And if you can see on the left side of the screen, there are some red tracks. It's trash, like cosmic litter in this prototype. And that's why it doesn't measure neutrinos. The main goal why this prototype exists is trying to understand how this technology works, how the detector working, and to make it clear to analyze the information what we get from it. So especially for it, there's existing detector control system. This is combination of more than 30,000 sensors that are located on the ProtoDUNE. And they measure some kind of information, for example, like temperature of cryostat, porosity, and pressure of the liquid argon, voltage between the wire planes, current and voltage on the electric field, etc. On the right side of the slide, you can see the model of this ProtoDUNE and some real-time values on this picture. Below, you can see that ProtoDUNE collecting two different types of information. One is wire plane readout, so it's information about tracks, particle tracks, and another type that is a detector control system database. So it's information about sensors and another important information about system. We need this data, both of them, to work with tracks. But the problem that DCS-DB needed for reconstruction had such high granularity. That's why we need data reduction in it to make it easier to analyze in future. So to start with my project, I'm going to tell you about original analysis that was made by Kevin Wood, whose plot you can see on the left side. He's made analysis where you could see in which time period beam was on or off. Also, there are some plots about beam moment, first graph, and the third graph, it's drift field, like an electric field. Also below this third graph, you could see like a grand red stripe of the stable and unstable time periods, where when system was unstable or unstable, and you could see the correlation between the straight like horizontal line of voltage with the stability of the system, and where we have drops of the voltage, there are some point stripes matched as unstable. It's important to understand that we're interested just in beam on information for the future. And for the further analysis, that reduction ignore remove all the unstable time periods information of the system. So to start with the analysis, I'm going to tell you about the electric field sensors like voltage and current. So in this run one, collection of all data, there was more than 50 plus million data points, and in original code that was used to build this analysis, they are using for loops for all data points. That was so long time periods to analyze it and to get the result. So on the right side of this screen, you could see the result of this analysis and the percentage of stable and unstable points. Also, I would like to tell you that values that are using for this analysis were averaged over a second, and cut or filter stable and unstable based on the hand-picked values of resistance and voltage. But it's not perfect, and you can see on the left side of the screen, you could see the voltage versus time plot, where you can see this straight vertical lines that are mentioned as blue, but we know that in this time periods value is changing and it should be like red. So my second step was to replace with pandas and numpy libraries all these for loops to make it faster, to optimize the code, and also try to the dimensional clustering of this database. So two-dimensional clustering is a type of algorithm that catches all this high-density clusters on the plot of voltage versus current, and using hyperparameters as a stable or a stable percentage. It's matching. We're trying to match original efficiency of the analysis. So also, I built plot voltage versus time, where you can see that this algorithm is not working well. Also, because you can see this. That's some blue points on the vertical lines where values changing, mention this appears as stable. So this algorithm is not working as well as I hope it could work. And the problem, there is no time dependency in this clustering. So that's why I'm using my own final algorithm. I'm trying to catch all the straight horizontal lines with all stable values of voltage. And for this, I'm using sliding window of variance and slope, trying with using hyperparameters as window width and variance, trying to characterize the lines of these values, because they have some kind of fluctuation inside it. We need to know that this line is straight on the like big time delta. So, and the result, final result of this analysis was intersection of stable voltage and current in one plot of voltage versus current. So it's important to understand that my final algorithm was working for the database voltage versus time and current versus time, and then we intersect them together. And on the right side of the screen, you could see that my plot has almost the original values of stable and unstable percentage, and almost the same picture of histograms of the where points are mentioned as stable. On the left side of the screen, you can see one of the two plots, voltage versus time, where almost all, or where every straight line are mentioned as stable, and all these vertical lines where values are changing are mentioned as red, so unstable. So in conclusion, I would like to tell you, I would like to say that the first step in my work, I'm trying to reproduce and understand the original analysis, trying to find out where I can improve it, where is problems of it, and how I can work with this data. The second step was to speed up code, like to replace Python for loops of pandas, numpy. Also, I also after that, I'm improved analysis algorithm using time dependence as a feature for the further analysis, and I hope that a new set of stable and stable time periods will make further analysis more reliable in the future. So I would like to thank IRIS-HEP and CERN for this very important, for this opportunity that they would have extremely important experience that I would use in my life. So thank you, everyone. Thank you. That's it.
Well, Volodymyr was mentored by Aranza. Aranza, are you here? You want to say a few words before?
Yes. Hello.
Yes.
Can you hear me?
Yep.
Yeah, I'm the mentor of Volodymyr, and he has really done a super good job in the LHCb experiment. So I think he's going to talk on it related to the trigger and a new algorithm that we are developing. So I just let him to talk.
Great. Whenever you're ready, Volodymyr.
All right, so thank you a lot for a great introduction. So my name is Volodymyr Svintozelskyi. I'm from Transcription National University of Kyiv. I'm the first year master student, and the topic of my project was the development of high-performance reconstruction algorithm for the detecting long-lived particles. So first of all, I'm going to give a small overview of the LHCb tracking system. Then I'm going to speak about the long-lived particles and especially about the tracking them within the LHCb. After this, I will move forward to the reconstruction algorithm, which I'm going to optimize in the next chapter. And finally, I will speak about the optimization results, actually. So let's move forward. So the LHCb is a specialized p-physics experiment located at LHC. So this means that it is applied with a certain Tarleton-Wald proton-proton collisions. And the three main detectors which are actively using in the tracking are the well or vertex locator, upstream tracker, which is located right before the magnet, and ski-fi tracker, which is placed right after the magnet. So there are a lot of major detector updates in run 3. So basically, we have significantly updated readout systems, trigger strategy, and so on. So basically, in some sense, we can say that we have a completely new detector for the run 3. And also, this means that we don't have a hardware trigger anymore within the system. And this means, again, that all the high rate trigger decisions should be done within some online machine. And of course, this online machine should support a very high detector output rate, which is like 40 terabit per second. So in order to handle this, there was proposed an innovational GPU-enhanced LHCb data acquisition system, which contains of roughly 500 GPU cards. This system is handle fully the HLT1 stage or high-level trigger 1 stage, and then the reduced data rate with some trigger decisions on the rate from 1 to 2 terabit per second are passed to the HLT2 stage, which is actually executed on x86 servers. So let's talk about the HLT1 stage itself. So the software, which is called Allen, or a high-level trigger for the GPUs on LHCb, is responsible for the HLT1 processing. So basically, it is developed using the CUDA-based framework. It is actually based on it, and it is fully implemented HLT1 stage. So this means that this software handles the huge input data rate, like 40 terabit per second, and make primary reconstruction of charged particle trajectories. It finds also the collision points, identifies some particles or hadrons or muons, displays the decay vertices, and also what is very important, it actually triggers on some interesting keywords, and therefore it filters the input data and selects only the interesting keywords. So So let's not speak about long cloud particles. So let's speak about some example, which is actually the lambda beta to lambda gamma decay. From the simulation, we know that 51% of these decays, of the tracks which correspond to these decays, has the UT and SCFI hits, while 37% of tracks has only SCFI hits, and therefore it is very important to be able to reconstruct such tracks, which are quality like a downstream track if it has like a UT hits and SCFI hits, and T track if it has SCFI hits only. And also, if we will be able to do this, we will have a lot of benefits, not only for the lambda beta lambda gamma decays, but for the similar decays, and also for maybe some non-standard model particles. So this is a very important thing which will be very nice if you'll be able to implement it. So this is like a global objective of my group. And again, it is a development of non-valid trigger lines, or trigger lines that triggers on the tracks that does not have valid hits. And constraints is obviously the output trigger rate and the algorithm throughput. So basically, we should be able to process the events fast enough, and also to somehow filter them just to reduce the output trigger rate, which is actually the idea of the should-given stage. How we can actually, for example, reconstruct the T tracks? Obviously, we can simply pass all the SCFI seeds that were reconstructed from the SCFI detector itself, and pass it as a T-track trigger line. But it will take a lot of trigger rates, output trigger rates. Therefore, we need to filter it, and obviously, the SCFI seeds that we are able to reconstruct consist of long tracks segments, downstream track segments, and T-tracks itself, but also in addition some ghosts. So this means that if you will be able to remove these two parts, then we will be able to significantly reduce the output trigger line, the output trigger rate for our T-track trigger line actually. So for the moment, there's already developed the long track reconstruction algorithm, and the downstream track reconstruction algorithm is under development. But the long track reconstruction needs some optimization, just to reduce the cost rate and reduce the output trigger rate, which it takes. And if we will be able to optimize this long track algorithm, we will reduce its trigger output rate, and we will make some space for another algorithm like a T-track line and downstream track line. And actually, the optimization of long track reconstruction algorithm is the objective of my project. So let's talk about the reconstruction of long tracks. It is performed using the so-called matching algorithm, which takes the track segments from the Velo and SCFI and tries to construct from these two segments some long track. So how it does this? First of all, it extrapolates both SCFI and Velo tracks as a straight line to some matching position. For example, in X coordinate, the matching position is placed somewhere within the magnet, and the exact matching position is derived per track, so it differs from track to track from the magnetic field parameterization. And you can see actually this on the bottom left image. While in Y coordinate, we have a matching position at the end of the SCFI, so we are basically extrapolating the tracks to this point. And then when we have a lot of SCFI tracks and we have a lot of Velo tracks, and for each of them we have some extrapolated points, we are making a loop. So basically we are looping over every SCFI track and trying to find the best Velo candidate for this SCFI track. And we are doing this by, first of all, filtering the Velo tracks by the slope difference between SCFI track segment and Velo track segment, but also by difference between these extrapolated points. And after this, we are trying to construct the chi-square for every resulting pair velocity phi. And using this chi-square, we are selecting our best long-track candidate. So now let's talk about this again, but now using some formulas. So first of all, we are filtering using the slopes of the velo and the ski phi. And also we are filtering by the distance between the extrapolated points. So this means that we have already 4 different parameters that we may optimize in future. Yep. So after this, we are trying to calculate chi-square using this long expression. And this expression also has additional 6 parameters like FDTY, FDTX, and these 4 right here. They are highlighted in blue. So in total, we have 10 different parameters that we possibly may optimize. And in the end, we have some additional chi-square cut, which is independent of all these calculations before, and actually it serves as a filtering of ghosts. But anyway, this is like an additional parameter. So in total, we have 11 parameters. So how to optimize all this stuff? So first of all, I need to mention that all the developments are done within the LHCb framework. And this means that some time of my work was spent just to learn how this LHCb framework works, just to learn how the Allen works, just to be able to launch at least something and so on. It's all is something like complicated things, but anyway, it's very interesting one. So anyway, we have 11 parameters that we should optimize, and one of them are independent. So basically, this is the chi-square cut. So I have split my like optimization strategy into 2 steps. On the first step, I'm trying to understand which parameters are interesting, which parameters may improve my situation somehow in either terms of ghost rate or in terms of efficiency. Because I simply cannot make all the possible combination over 10 parameters, as it will take very much computational resources. So I'm selecting, and I'm doing this by sweeping only on one parameter at a time and check its own impact. So basically, I'm selecting one specific parameter, then I multiply this parameter by some factor in a range from 10 to the power minus 2 to the 10 to the power 2, and trying to understand how exactly this parameter affects my efficiency and cost rate. And after this, when I will have some information about each of the parameters, I will select some parameters that might be useful for me. Yep. So they are able to either reduce the cost rate or increase the efficiency. And then using this selected parameter sets, I will try to make the all possible combination loop and finally make a decision on these 10 parameters. And finally, on the step 2, I will try to simply optimize the chi-square cut by simply sweep on the some possible values. So let's talk about the first step. This is the plot total efficiency versus total cost rate. Each point represents some specific set of these 10 parameters. And actually, each line represents a sweep on one specific parameter. So, for example, you can see that blue line. When I'm sweeping on the blue line, I can get, which corresponds to the DTX parameter, I can get a very low efficiency. But obviously, the efficiency like 10% and cost rate like 20% are not really interesting for me. So let's zoom in this plot to the interesting region. So here we go. This is the interesting region with a relatively high efficiency and low cost rate. And big black point represents the default set of the parameters. And from this plot, I'm able to select the interesting parameters. So for example, this brown curve that corresponds to the FDTX parameter may slightly increase the efficiency, while the gray line may significantly, for example, reduce the cost rate, which corresponds to the parameter dy-2. So using this approach, I'm selecting the interesting parameters, and the number of interesting parameters is 6. And you can see them on this slide. And the next step is to try to create all different combinations of these parameters. And on this plot, you can see the results of this all different combinations. which red point corresponds to some specific combination. And therefore, I can say that it's possible to either increase efficiency by roughly 1%, but with increasing the ghost rate by 3%, or to decrease the ghost rate by 1% with almost no loss in efficiency. So, but to characterize all this stuff in more detail, I need to select some characteristic points with, for example, best efficiency, best ghost rate, and some intermediates. So here you can see these points. And then I need to compare for each of these points their efficiencies, their ghost rate, their output trigger rate, and their throughput. So basically here you can see all this stuff. So here's the efficiency, here's the output trigger rates, and throughput, and all system throughput. And on the top part, you can see the exact parameter values for each of the sets. So basically on the first column, there are the default values, and every next column, there are multipliers on which these default values should be multiplied by to get this exact set. So we have 4 different columns, which corresponds to each of these 4 points, and then we need to make a decision. So which one is the better for us? It was decided to move forward with the best ghost rate set, which almost does not change the efficiency, but reduces the ghost rate by 1%. And this means that we also reducing the trigger rate, the output trigger rate, which is actually the idea of all this optimization. So we have completed the step 1 when we have optimized the first 10 parameters. So now let's optimize the last parameter, which is max chi-square cut. So on the top left image, you can see the distribution of different tracks, or the chi-square of different tracks. So the yellow line corresponds to the ghost tracks, which are not correspond to any real particle, while the blue line represents some tracks that correspond to some real particles. And actually, from this plot, you can see the number of tracks of each type. While on the bottom left corner, you can see the fraction. So for example, starting from the max chi-square cut equals to 2, we have more ghosts than matched tracks in our cocktail, if I can say so. So it's obvious from this plot that we may optimize this maximum chi-square cut, and we definitely should do this. And let's try to optimize the chi-square cut by simply sweeping in some range of possible values with some step. And again, try to plot the results on the same efficiency versus ghost rate plane. And here you can see the results. So again, as in previous case, I'm selecting a few interesting points and then making a detailed table with rates and throughput and everything else for these 4 points. And from this table, we need to again make some decision. So with which max chi-square cut we should move forward. And it was decided to move forward with a maximum chi-square cut equals to 2.0. So now let's compare our newly optimized matching algorithm with non-optimized one. So this is the efficiency plots versus some variables like eta, p, or pt, and so on. And from these plots, you can see that efficiency almost have not changed from our optimization, or to be more precise, it have not changed within the error bars. So it's actually the same. While the ghost rate is reduced a lot. So this is the result of my work. So to summarize all this, I'm going to repeat this once more. So I have optimized the matching algorithm for long tracks reconstruction. The results are almost not changed at efficiency, or to be more precise, the same within the error bars, and reduced the ghost rate by 1.56%. And one of the most important things, I guess, within all my presentation is that these results are already implemented within the official LHCb software, or to be more precise, within the Allen software, as my merge request was accepted, and therefore all these results are already available. So the next steps are the implementation of the T-Tracks trigger lane and possibly its optimization. So I want to thank organizers for this amazing opportunity to work on the projects which are really interesting for me, to work on the actually something which corresponds to my specialty. I want to thank to my supervisor for great support during whole project development and so on, for very great advices and everything she did for me. And also I want to thank to my group of Valencia group that make a lot of very useful advices and a lot of great discussions. And finally, I want to thank everyone here in this Zoom room for an attention. So that's all.
Thank you, Volodymyr. Any questions? Oh, Gordon, you have your hand raised?
Yeah, Volodymyr, a great talk. I'm doubly interested in this because I'm also a huge fan of long-lived particles. But just sticking to some of the math questions, it looked to me like the whole form was a continuous function that you were trying to optimize. Did you give any thought to just using a fitting framework built on, PyTorch, JAX, or TensorFlow or something like that to try and optimize for the best efficiency and smallest ghost rate, as opposed to doing the parameter scans, which it looks like has been more your approach here?
Yep, that would be a great approach. So thank you a lot for this question. But the point is that I was working within the OHCB stack, just to be sure that everything that I will change will also work within the official release of the Allen, and it will work during the real data taking. And therefore, each iteration of this optimization needs the restarting of the whole Allen software with whole analysis and so on. So it's not really simple to I don't know, to include it within some greater frameworks of optimization.
Okay.
So it's a manual job.
And then I had one other quick question. Normally, chi-square has a fairly specific probability distribution definition. By modifying all of these, I guess it was 11 parameters besides, or maybe it was 10 parameters besides chi-square. Was there any attempt to understand whether or not the chi-square was still actually a chi-square at the end, or fit to that, so that, you said the default cut was 2.5, I think, and that's because that corresponds to a probability in the distribution. Did you ever look at the chi-square to see if it was still behaving like a chi-square?
Oh, to be honest, not really. But what I can say is that within this framework, this chi-square was constructed like this, just to be able to compute it quickly, because it's all running on the L31 stage. Therefore, we cannot make a lot of computations here, and we need to merge two text segments into one long track line, and therefore, yeah. Therefore, it
makes sense.
Looks like this. And what I really know is that initially these parameters were chosen just by chance, so nothing really. And also that was, and also actually this algorithm was transferred from the SLT2 level from the previous OHCB runs. So therefore, maybe some of the parameters initially was set to the same. It wasn't the SLT2.
Sure. Yeah, you could approach that by just throwing everything into a machine learning network, ignoring this particular thing, see how well you can do, and then you can tell if maybe there's some other thing that you could do with this simple calculation. There's a piece of phase space which is even more efficient and better at rejection. But look, this is a great talk and a lot of work. Pretty impressive. Thanks a lot.
Yeah, thank you a lot for the questions.
All right. Any other questions? No. Good. Good talk. Gordon, you were one of his mentors. Do you want to
Yeah.
introduce?
Yeah, I just want to say it was an absolute pleasure working with you, Haoran. You're an undergraduate here at the University of Washington, where I am. So I've known you for longer than this this particular project. But the problem that he was attacking here has been a long-standing one within IRIS-HEP and the ServiceX delivery, which is how to modernize some of our CMS our CMS tooling. And he was incredibly brave because he tackled this without actually being a member of the CMS collaboration. But thanks to CMS's open data policies, we were actually able to get at the information and also the folks that we needed. So really, really nicely done. And take it away.
Okay, thank you so much for the introduction. And hi everyone, my name is Haoran from University of Washington. And my presentation will be on enabling support for MiniAOD transformer for ServiceX data delivery service. And my mentors are Professor Gordon Watts, Dr. Alexander Held, Dr. Oksana Shadrova, Dr. Ben Gelsky, and Mr. Prophet. And okay, so I would like to start with just a very brief overview of the content I will be covering. I will start with an introduction of ServiceX and then move on to introduce functional ADL, which is a data extraction tool used for ServiceX. And the main part of this presentation will be discussing the architecture of functional ADL. And finally, I will discuss the result and the conclusion of this project. So let me get started. So what is ServiceX? It is a data extraction and delivery system for high-energy physics event data. And what it does is to locate, extract, filter, and transform the data. So this image here shows a basic workflow of ServiceX, and you can learn more from this website link below. But in summary, it starts by translating functional ADL language, which I will explain in the next slide, into QREST language expressions, or CASL for short. And these CASL statements will then be translated into some sort of C++ or Python code. But in this project, it will be C++ code only. And the CPP code will be ready to run on the file that stores the high energy physics events data. The file which we will run on will be looked up by the DID finder. And the product after running the CPP code can be stored in the kilometer file formats, such as a pandas data frame or awkward array for easy analysis. And as I mentioned, the ServiceX workflow starts with funcADL language, and it is a SQL-like declarative query language, which means you tell it what you do instead of how to do it. And it extracts and filters the physics objects from a file and returns them in a kilometer format, such as pandas data frame again. And here it's just a snippet of an example funcADL code that I took from the ServiceX documentation website. So what it does is that it selects the jet collection from the Atlas XAOD file, and then it selects the PT attribute of each jet in the jets collection and divide each of them by 1,000. And then, as PandaDF does, is to store the result data as a pandas data frame. You can do a similar thing to convert it into an awkward array. And the value function basically tells the language to execute the query. And before this project, the funcADL has already supported flatroot file, Atlas XAOD file, and CMS ROM1 AOD file as its backend. And this project enabled CMS ROM2 MiniAOD file as its backend. And okay, for the next few slides, I will be talking about the architecture of the funcADL. The first step funcADL does is to represent the query that the client writes as into a Python abstract syntax trees, or just AST for short. AST in this case is a tree representation of the query, and it conveys the kind of the whole structure of your query instead of just a single string. And here I have this kind of a comparison between the query that the client wrote and the AST representation of this query. So as you may see, AST takes the query string and sort of like breaks it down into the components and represents its structure and logic. So what I mean by that is, for example, AST will know that select many is a function that is being called on the F single, which is a event dataset object, so on and so forth. But in case you have not noticed, one of the functions is named CPP code value, which does not appear in my original example query. And in fact, the CPP code value is a class that has different fields which are used to store the proper information that will later be transformed into the C++ code at different places in the CPP file template. The muons in my example query code is the name of an object called event collection specification. And what it does is that it specifies information like which header files I need to include in my produced CPP files, or the type of collection I need to iterate over. And this function, get collection here, basically puts all the information that are stored in the event collection specification into the CPP code value. And then it will be processed later in the ST node. Okay, so after the query is represented by a Python AST, an executor object will be created. And in this project, it will be same as MiniAOD executor specifically. Similarly, if you are running a function ADL against Atlas XAOD file, then it will just be Atlas XAOD executor, etc. So what the executor does is to simplify the AST on the last page through the function called apply AST transformation. And then it will traverses through the AST and turns each AST into some sort of CPP code. And the main work is to traverse through the AST, which is done by an object called query AST visitor. It is a subclass of AST.node visitor, which is a existing Python method of AST package. And what it does is to walk through the AST node. And as it does so, it will extract and accumulate the needed C++ information, such as type definitions or the variable declarations. And then the accumulated CPP information will then be stored in different attributes of the object called generated code. And some of its attributes are shown in the graph. Oops, sorry. Okay. So the next step is to inject CPP code into our CPP file template. And we do that through the function called write CPP files. Here I have part of the code from the write CPP files function, which injects the class declaration code into the template file. So basically the get visitor object function returns the query AST visitor object I mentioned in the last page. And we then get the class declaration code information, which are stored in the query AST visitor, and then we place it into our information map. And then by matching the names of the keys of the map, in this case, it's a class declaration with the template. Oh, sorry for the red square not in place. We can, by matching the name of the keys of the map, we can place the corresponding values, in this case, the code for the class declaration, into the template as a red place. And after completing the C++ template with injected CPP code. The CPP file will be ready to run as part of the ED analyzer, which are part of the CMS software. And there's modules that allow read-only access to the MiniAOD. You can learn more details about the ED analyzers through this link right here. In this project, the ED analyzer will be run inside the CMS software container, and it will follow the commands given by the shell script called runner.sh. And in this project, we use the version 767 for the container. And for the next section, I will discuss about the results and conclusions of the project. Here I show the initial query code that the client wrote, and its corresponding generated CPP code snippet that will run on MiniAOD file in the backend. And here is the a visualized result root file after running the query on a test MiniAOD file with only 20 events. And this root file can be easily transformed into other preferred columnar data formats, such as awkward array or pandas data frame. So the way we approach these projects is to first study the structure of the functional ADL code and study the difference between CMS AOD file and MiniAOD file. And also we write in ED analyzer in the container manually and study the correct form of it that will return the desired data. And we then try to generate the CPP files through funcadl, which we wish to match the manual files that we wrote in the container. And the ED analyzers used for processing data are very alike for AOD and MiniAOD, but there are a few differences that we had to spend an amount of time on. And here I have one of the examples, which is the ways they access the data. For CMS AOD, one can use get label function, which directly takes the string as its module label. But it doesn't work for MiniAOD in this case. Get label is a deprecated function for run2 MiniAOD file. So instead, we use get token function, which cannot take a string directly as its parameter. Instead, we take this object of type edGetTokenT as its argument. And this argument has to be declared globally in the CPP file, and then it has to be initialized with its input tag in the constructor. And that makes the implementation a little bit harder. And so the general idea we solved this issue is to store those CPP codes that are for MiniAOD specifically into an attribute of the CPP code value through this function called getRunningCode CPP code value. And again, as a reminder, the CPP code value is a class that stores the desired information that later will be transformed into CPP code at different places inside the CPP file template. So this attribute of CPP code value will be empty when we run the CMS AOD file. But when we run against the MiniAOD file, it will contain the desired code that we wish to run. And there are just a lot of other different factors between AOD and the MiniAOD files that we looked into, but I cannot cover all of them in a timely manner. So sorry about that. So the future work on this project, we could try to enable the support for more high energy physics data formats, such as nano-AOD. Also, we can add more method types, so more functions of ED analyzer will be supported by functional ADL. And that, excuse me, and that wraps up my presentation. And at the very end, I would like to thank all my mentors, Professor Gordon Watts and Dr. Alexander Held, Dr. Oksana Shachura, Dr. Bengalski, and Mason Prophet for their enormous help on this project. And I would I would also like to thank the generous help from Beniano Kundu, who used to work on enabling the CMS AOD file for functional AOD previously, and Dr. David Lange, who helped me answer a lot of CMS related questions. And thank you for listening. That's the end of my presentation. So yeah, thank you.
Nice talk. Any before you get started, I think some of your mentors are.
I'm here. Yeah, I'm here.
Okay. You wanna introduce Bohdan's work?
Yeah, absolutely. It was a great pleasure to work with Bohdan on the project, and thanks for all the support in terms of like logistics from the fellowship. So the project was about genomics and the wastewater, and that's actually the quite a new frontier in the genomic research. The way it is, so we have figured out how to do wastewater using the PCR, but the sequencing based wastewater, it's absolutely the new frontier. And then that was really exciting to work with Bohdan. So the project is actually about benchmarking. So basically benchmarking of the methods for the lineage detection in the wastewater. So benchmarking is the study in order to find the best bioinformatics method able to provide the best accuracy. So which Bohdan will talk about.
Thank you, Sergey. The topic of the presentation and my work is benchmarking of methods for SARS-CoV-2 lineage detection in wastewater, as Sergey has mentioned. This work for me has been done remotely, but the lab I'm working for is situated in LA. It's Mango Lab, University of Southern California. And I work directly with Sergey Knyazyev, who is the author of one of the methods I will be talking about and how I have benchmarked. Let me introduce you what is wastewater monitoring and how it already has proven itself successful or that it has potential. So there were multiple studies which show that basically wastewater monitoring for COVID sequences in wastewater proved to be effective in determining the trends in disease spread. And also the problem which I am working on in the lab is using wastewater material and particularly sequencing data extracted from it to detect new variants of COVID, which would be helpful in order to protect ourselves in case some variants some dangerous variants emerge. And the issues are that it's difficult to identify lineages at low concentration of them in wastewater. And also tools might detect some lineages which are not seen in clinical samples. So it's like tools creating imaginary samples of COVID, imaginary variants. So the proposed plan was to generate a benchmarking data set which simulates the output of Illumina sequencer which is a machine for turning DNA material into a data set of reads, which are pieces of virus DNA. And then we create those mixtures of reads. This is our data set on which we then benchmark our tools. There are 3 tools we're tasked to benchmark. The tools output new detected variants, and then we estimate their accuracy. More concretely, our pipeline look like this. We set up environments for every tool because some of them really take a personal approach to them. Some of them use some old libraries and stuff, and old C++ compilers. Then we generate sequencing reads. Those we generate from variants found on the main COVID database on the internet called GSAID. Then we align those reads, creating these mappings of these reads onto the variant sequence of the virus, putting them in the particular places on the sequence. And then we're running tools, saving the outputs, and in order to estimate the accuracy of the undetected variants, we need to do some processing and alignment of the outputs. The challenges I encountered were that the tools were poorly supported, incomplete documentation, or the tool can be old, which is also a problem. We need to install some additional software and stuff, and it might not be obvious what to install. And also I'm working remotely, so less opportunities to communicate, and it has some downsides for me personally. As a result, we created a bunch of mixtures of reads, like 8 data sets, which we then used for benchmarking. And I tried a number of different approaches to setting up my workflow, working with all the abundance of tools I have on hand. I also forked and used extensively the repository of one of my mentors. He's also a co-creator of one of the tools I benchmarked, ClickSNV. And these scripts of his were very useful throughout my work. This is the table which we get for now. we can see that the variants detected for each of the mixture are non-zero. So mixtures to find some variants in the mixtures. We can see 7, 5, 2, 3. And by the way, these mixtures have both 5 variants in them. So we have some overshots and some too small amounts of variants detected, but precision recall is 0. And it needs some further research because we can in the future use multiple sequence alignment to improve the scores, because you know, there are these phenomena as insertions and deletions in genomics. Also, we need to make sure that the sequences start at the same kind of place, same at the same position on the genome. This should be really accounted for, so we'll look into that. And also, we can benchmark other tools. Some papers of my mentor have other suggestions. And the pipeline, the proposal contained the suggestion that I should use SnakeMake in order to make my workflow like easier for me, and the results reproducible and easily expandable, like if we want to benchmark some other tools later. And I tried using it, but ended up thinking that it's not really worth it. And we decided in the lab that we will look into other pipelining tools. We have one on the radar. Thank you. It's been a pleasure to be a part of this project, and I hope to bring more impact in the future. And thanks for the IRIS-HEP for the opportunity.
Sorry. Thank you, Bohdan. It was a nice talk. Any questions for Bohdan?
So, hello everybody. My name is Volodymyr Shabanov, and I am a student in Karazin Kharkiv National University. My mentor was Dmytro Horyslavets, and this study is related to the role of myocardin transcription factors in the smooth muscle cell gene program. So, as the name of this project implies, we analyze the effect of expression of myocardin and on other genes in smooth muscle cells in the human coronary artery using the RNA sequencing method. Smooth muscle cells are very important building blocks of human bodies. Smooth muscle cells tissue is found in the walls of hollow organs, including the stomach, intestines, bladder, and uterus. In the wall of passageways such as blood and lymph vessels, and many other places. Because of wide variety of organs that contain smooth muscle cells, there are wide variety of smooth muscle cell phenotypes. Smooth muscle cells can be differentiated into more specific phenotypes and they and through their life cycle, they can change their phenotype. The change can be induced by internal processes and also by environmental cues. The process of inducing phenotype change in smooth muscle cells is controlled by many transcriptional activators, one of which is myocardin. Studies found that disruption of myocardin activity can lead to abnormalities in development of organs, such as the organs of cardiovascular system. And so understanding the effects that myocardin has on these cells is very important for many areas of medicine. So, to study this effect, the effect that myocardin has on cell, we need to look through the genes that are being expressed in this cell. And to study the genes, we need to study the number of RNA molecules that correspond to the gene that were expressed. And so for the study, the smooth muscle cells were treated with adenoviruses that express nothing in the control group and overexpress myocardin in the treatment group. This was done so the cell reaction of being infected with the virus would not be taken into account as a possible effect that myocardin has. In the treatment group and the control group, there were 4 samples. After the treatment, RNA was extracted from the cells and sequenced, which means can be converted into usable computer data. After that, they were uploaded on the public NCBI server. And from this point, I began my project. Firstly, this data was downloaded from this server, and I did it using the command line because of the sheer size of these files meant that I cannot download them from the web using my browser. I did it using the SRA toolkit software. After the downloading, I needed to look through the data to see if it was usable for the study. I used software which could analyze the complete set of RNA sequencing data and determine the overall quality and usability of this data. There were a lot of parameters that I have checked, but I think this is the most descriptive one. This slide shows the flat quality score of the samples depending on the size of the RNA segments. Flat quality score is a measure of quality of the identification of nucleotides generated by automated RNA sequence. The flat quality score above 30 means that the error could be found only in 1 out of 1,000 nucleotides, which is pretty good results. The result of this analysis supported that the data which were analyzed contains RNA segments of mostly above average quality, with which further research could be carried out. After that, we needed to quantify these RNA segments. For this part, as you can see on this figure, I had to align the data to corresponding parts of the reference genome. For this part, I had to download the certified release of the human genome. And after alignment was done, the reads which correspond to their position, in their position to the position of the gene can be counted. In the end, we got the count matrix, which contains the gene ID and the number of times which express for each sample. You can see this kind of matrix here. For this part of the project, I had to import the data into the R programming language because it has a lot of useful packages for RNA analysis. You can look at the packages I used on the right. After that, one of the key quality indicators the data has is the library size, which reflects how much of the RNA segments has been covered with sequences. The library size means the number of reads that were sequenced. After getting the count matrix, I created this bar plot, which visualizes the size library of reads that each sample has. As you can see, the samples are color-coded. Green is the treatment group, and the purple is control. The bar plot has the red line, which indicates the mean size across the samples. The samples from the control group and the test group have similar size, and also the mean library size is about 35 million reads, which is pretty good. It's above average because the accepted library size for the human genome is usually considered to be 20 million reads. After that, to make sure the treatment that we studied had a significant change, we made a principal component analysis and made a plot on its results. For the analysis, we used, we can see that 90% variance recorded on PC1, which means that the treatment was the main reason for the difference between the samples from the test group and control group. And PC2 visualized how did the samples in each group vary between themselves. And we can see that 3% is negligible. The variance is very low, which again indicates that the samples had the same treatment and had similar results from it. After that, we did a differential expression analysis, which means taking the normalized read counts data and performing statistical analysis to discover quantitative changes in expression levels between experimental groups. For example, we use statistical testing to decide whether for a given gene an observed difference in read counts is significant, that is, whether it's greater than would be expected just due to random variation. For this part of the analysis, I used the generalized linear model. It made corrections on the size of, on account of size of sample library, so no genes would be overrepresented, and it checked whether the sample was from the treatment or a control group. It's condition part of this formula. And later, it was checked using the FDR, false discovery rate value, to filter out the random noise. We can see, you can see this, in the end, I got the table that contains the genes and their corresponding statistics, and you can see this table here. We can see that on each gene, the mean was calculated, the logarithm of change that the treatment has brought, standard error of the logarithm, and the results of the significance test. Arguably, the most important values of this file is log to fold change and p-adjusted values. which shows the effect size and the significance of change which was brought by the treatment. From this data, I could figure out how many genes were upregulated and downregulated. It was calculated that in total there are 5,450 genes were downregulated, and there are 5,951 genes that were upregulated after the treatment. After that, I had done the functional analysis of the data, with which the most prominent genes were analyzed and put in the gene ontology group that was in their respective gene ontology group. In this figure, we can see which group of the genes were upregulated, colored blue, and which were downregulated, colored yellow. As you can see, also you can see how much a change was brought by the treatment. The most affected gene was ACTA1, which encodes actin alpha-1 protein. When expressed, this gene causes expression of several other myogenic genes, which are essential to muscle formation. Other most upregulated genes, such as cardiac muscle alpha-actin, play a key part in the muscle formation and differentiation. Other notable genes that were upregulated was troponin C type 1, whose main function is regulation of the ATP activity, and retinoid isomer hydrolase, which mainly affects the cell ability to react to stimulus. Also, myocardin gene was upregulated too, but because of the nature of the study, upregulation of myocardin was expected. The most downregulated genes were a part of the genes related to cellular response to biotic stimulus, interleukin 36 receptor antagonist, and which mainly act as anti-inflammatory protein, and granulocyte colony stimulating factor, which contains the response for apoptosis or programmed cell death. Maybe this change was caused due. The cause of this downregulation is not completely understood for me, I guess. I think because the cell, because of the differentiation of the cell could not have created a sufficient response to the infection with the virus. So the proteins that contain the response for this virus were downregulated. This can be a subject for further studies. In conclusion, I can say that myocardin is extremely strong regulator of other genes. It was seen on the PCA table. Many of these genes play a key role in muscle tissue formation and differentiation, and the study produced a list of genes candidates for further study. I have learned the full bioinformatics pipeline of differential gene expression analysis, from downloading the raw data to the tools of the gene analysis. Thank you all very much. It was a pleasure being a part of this program, and it was a pleasure doing this project. Thank you for listening.
Thank you, Volodymyr. Any questions? I see I see your mentor is connected, Dmytro. Do you have anything you'd like to say about?
I think it was great. As Volodymyr said, the main goal of this exercise was for him mainly to train how to do the analysis. And given that he had no experience in this area before, I think he did a great job. He worked very independently. I barely helped him. I was just giving him instructions and he followed and he did everything well. So yeah.
Okay.
It was pleasure to work with him. Yes.
Yeah. So the title of my project was sequence root extraction from counting the Bruijn graphs. My mentors are Andrii and Haoran from ETH Zurich and Alina Frolova from Institute of Molecular Biology and Genetics of Ukraine, of NASA of Ukraine. A sequencing cost is decreasing the amount of biological sequencing data in public repositories is growing exponentially. This creates a need for indexing of and searching raw sequencing data at petabase scale, which means that it is like the scale is 10 to the power of 15 base pairs. So for comparison, the entire human genome length is about 3.2 billion base pairs. So the repositories of sequencing data are really huge and they typically consist of lots of samples. And the compressed representations of graphs are a promising technology for efficiently indexing k-mer sets and performing search and sequencing data. The main concept which is used in this project and implementation is the Bruijn graph, which is a directed graph which represents the overlaps between sequences of symbols. A colored Bruijn graph is a generalization of ordinary Bruijn graph to distinguish multiple samples. It basically means that each node can be like colored, which means it can have a label which represents from which read it was originated from. The count in the Bruijn graph is a notion generalizing annotated the Bruijn graphs by supplementing each node label relation with some additional integer set attributes that can be those k-mers counts or positions in the original sequence. The coordinates in this case basically encode reads as graph walls. And counting the Bruijn graphs concept was proposed by Andre's team. So here you can see like the example on the right side. You can like take a look that each two adjacent nodes overlap by two symbols, and they are colored in the colors of those example reads from which the graph is constructed. So the task sounds can be formulated as follows. So given an annotated count in the Bruijn graph and the query sequence, we want to get the set of all input read sequences that overlap with the query. This is supposed to be implemented as an option in the metagraph framework, which I was talking about in the first presentation. Metagraph is a versatile framework for scalable analysis of extensive sequence repositories. And it's worth saying that both graph and annotations are stored in a compressed format in this framework. So let me show you how the implemented algorithm works with some example. So consider like this simple graph on the left side. Let's say that we pass the sequence ATGC as an input query, which corresponds to this node, input node. The first step of the algorithm involves traversal of the graph forward and backward from the input node. This results in constructing adjacency lists for these nodes. After that, for all visited nodes, the annotations are fetched, and the annotations are stored in a compressed format, as I said before. That means, and the annotation contains like two parts: the position of this k-mer in the input read, in the initial read, and its label, basically. So here at this picture, the colors of the nodes represent those labels of sequences from which the graph was constructed. After that, as we have those annotations and like adjacency lists, we can find the walks into graphs that includes an input node, because each walk has its initial node and terminal node. So we just reconstruct those paths in the graph and just reconstruct the read sequences and return the result, as a result. And you can see that indeed the input sequence which we passed is actually present in both these sequences. If, for example, the input sequence had length more than the k-mer size, than the length of those nodes k-mers, then the algorithm will work for each k-mer of the input sequence. We also have plans for further work because first of all, because this implementation, the algorithm, this task can have applications, useful applications for some real world data. Here written some possible use cases. For example, you can search, it may be used for search of unknown DNA in some samples, or for some purposes in single cell sequencing. And also, we would like to improve the algorithm to make it more, to make it scalable, because, which means like basically 2 points. The first one is to implement local graph decompression, which means that the decompression of the annotations will be done during the graph traversal, so sequentially, instead of just traversing the whole graph and then fetching the annotations for each label. And that will result in more, that will basically mean a more efficient algorithm, especially on large graphs, because a lot of redundant work will be omitted, which are now present in current implementation. And the second point of the scalability issue is the batch mode, which means that it would be good that if there will be an option to process multiple input sequences simultaneously. So yeah, that's it. Thank you for your attention, and thank you for the opportunity to participate in this fellowship.
Thank you. Nice talk. Oh, your mentor is connected. Andrei, do you want to say a few words about Dmytro's work?
Yeah, sure. I'm happy to share. Yeah, I mean, we had great luck with Dima. He looked. I mean, we threw a quite complex problem at him. So, and he worked on it quite independently. I mean, first he had to somehow tackle a code base of over 100,000 lines C++ code, and now he worked through that and managed to add a feature. And yeah, now we work to scale that even further. I think that the complexity comes from the fact that we're working on huge data, and that the graphs that looked so neat in Dima's presentation, I mean, can have up to 1 trillion nodes, which complicates things. And that means you need to jump through many hoops algorithmically to locally decompress information without spending too much memory. And yeah, I think he has conceptualized it very well. And yeah, we're looking forward to keep working with him on this. So I think he agreed to also continue to work maybe for his master thesis in the topic. We're very, very happy.
Connected. Savannah, you want to say a few words about Ameya's work?
Sure. Yeah. So Ameya worked on a really interesting project. The first one, at least as far as we're aware, of using equivariant networks for charged particle tracking. Also the first, again, at least as far as we know, application of like rotation equivariant GNNs to particle physics. There's been some work on Lorentz invariant networks, but this is a bit different. So it was really interesting project. He did a lot of great work figuring out like how do we actually enforce this kind of symmetry, and then how do we kind of understand the results. So yeah, it was a pleasure working with him, and I'm excited for everyone to hear the talk.
Thank you.
Hi everyone, my name is Ameya. I'm a senior at Pilani, Goa, where I'm majoring in physics and computer science. My project over the summer was on developing equivariant graph neural networks for charged particle tracking, and I was mentored by Dr. Andrii Len and Dr. Svintozelskyi. Produce tens and thousands of particles, and these particles interact with specialized systems called trackers. And in doing so, these particles leave energy deposits called hits in these trackers. So it's something like the puzzle where you connect the dots to see the bigger picture. However, are unsuitable for high luminosity conditions that we're going to see at the LHC. So this prompts the search for better techniques. Graphs are these data structures that can capture the sparse nature as well as the relational structure of physics data pretty well. Tracking data, as it turns out, is quite naturally representable in a graphic format. If the hits are identified with nodes, any track segments between these hits can be represented by the edges in this particular graph. To work with graphical data, we use a class of deep learning algorithms called GNNs, or graph neural networks. It has shown that GNNs can be really effective for the particle tracking problem. Now coming to really, summarizing what the project is about, alternative models for the tracking problem. As we will see, this particular problem has some inherent symmetries which we can exploit. Now, very closely connected to the concept of a symmetry is that of equivariance. A function is equivariant under some certain group G if by transforming its you transform its input, and that leads to a transformation in the output by the same group action. Now, just to make the concept more clear, you can look at this really silly example on the right way here. Rotating the image after applying some sort of color inverting function has the same effect as first inverting the image and then rotating it. So this color inverting function somehow is equivariant under the group of image rotations. Now, models that incorporate these symmetries through equivariance have been shown to outperform their non-equivariant counterparts. So what they do is, apart from telling the model about a certain background knowledge, which we call inductive biases, they also have the potential to reduce number of trainable parameters, or even the inference times these models take to produce outputs. So if you want to look at an example in high energy physics, a Lorentz equivariant top tagger could achieve a state-of-the-art accuracies and background rejection rate as compared to a non-equivariant tagger. For this particular problem, we're using the track ML dataset. So each event in this dataset contains the 3D hit position, that is the X, Y, Z coordinates for the hit, as well as the truth information about a particle which generated that particular hit. To simplify the graph construction to those which have a transverse momentum below a certain threshold, or that in a particular layer, there's only one hit per particle. And finally, now this basically just gives you all the nodes in the graph. Now the graph, the edges on the graph are constructed if a particular edge satisfies constraints on these two geometric quantities, Z0 and phi slope, which you see here. Yeah. And the the constraints are determined via just a hyperparameter search. And here on the right, you have an image of a hit graph for one event, which is constructed the detector. You can sort of make out the outline of the detector, and that is, it corresponds to actually a particle traversing any two nodes that the edge connects. Typical GNN pipeline for a tracking reconstruction problem consists of 3 steps. There's graph construction, which you already covered. There's edge classification and neural network that produces rates for each of the edges in the graph. Now, people study all sorts of optimizations which can be done at any form of this step, but as I said, this work particularly just focuses on step 2. So if you see the image for the pipeline here, we just focus on the second step here. Now coming to really what the project was about. The first step in this project was just to take a really long detector on the slide at the bottom right. And if you just look at it stare at the image for long enough, you'll see that it's pretty much symmetric about the beam line. That is this particular arrow that's pointing outward over here. And this sort of symmetry proof orthonormal rotations, which are in the XY plane, or rather, now that's one symmetry in this problem which we spotted. And now that we've done that, it might seem helpful to somehow use this knowledge and give it to the neural network before it can even begin training. And that's pretty much the motivation for why we want to study equivariant models. So we spot that there's some symmetry in the problem. We identify that symmetry and then tell the network, hey, like there's this particular symmetry that you might want to look out for, or rather just stick to functions that follow this symmetry because physics tells us that's something that's viable. Now, another thing which we can consider here is this. Most physical laws are equivariant to 3D rotations, that is rotations in 3D Euclidean space. So it might also be worthwhile to look at SO3 group symmetries. And so that is also what we do in this particular project. In this particular slide, I've just added this image here to further. Left is a plot of the hit graph in the XY plane with the two edges again shown in blue as previously. On the right, I have the same hit graph, but I've rotated it about the z-axis by 60 degrees. In the positive direction. If you just look at these graphs and give it like a cursory visual for these differences with a lot of effort. Since these graphs are thus more or less symmetric in the XY plane, this sort of gives us the motivation to maybe tell the network about this particular symmetry that these graphs have. Now, the second step in this project was building the GNN itself. So now we've identified the symmetries, we can start building the GNN. Another reason for this is the choice of symmetries you want to study sort of guides the way your GNN should look like. So it's already into Euclidean symmetry groups. And they're inspired by the Lawrence set architecture, which by Gong et al., which I think early last year. The inputs to the GNN are the head graphs, which I showed earlier, the vector. So 3 components of the feature, the distance, or rather the length of the edges, are not SO2 or SO3 equivariant, so they can't be used in in whatever computation the model is performing. So now that begs the question, what can the model use? And how are you actually going to include these symmetries? If you look at that, so consider this really tiny graph with 4 nodes and 5 features. We expect this graph to be equivariant under rotations, for example, in some sort of permutation invariant function like a sum, which I've used in this case. These features are transformed by rotation. The transformation, that is the rotation, propagates like so. All the inputs to the MLPs are transformed. So you have Rx's which are input to the MLPs, and these outputs are then aggregated. But that's pretty much where it ends. And if you look at this particular aggregation closely, it's not equivariant. Because if I aggregate the features and then rotate them, I can't obtain the same output as which I get from doing this particular operation, which I've done. So that basically goes against the definition of what a function being equivariant means. So there has to be a better way. And to do that, we modify the message passing formulation itself. If now in itself, we pass the norms of the feature vectors, differences. So if you're looking at node 1, we pass, XMLP. We can generate these messages, which are labeled by MI. So there's M2, M4, M3, and so on. And these messages are then used to weight the difference, Xi minus X1, in the aggregation step. And this gives us a new updation scheme, so to say. Now, the good part is this method is equivariant. Because these messages, firstly, these messages are invariant under rotations. You can look at this really tiny. Any rotation matrix, which you apply before aggregation can sort of be factored out. So these rotation matrices, so to say, in quotes, can be factored out. This ensures that the order of operation doesn't matter. So basically, whatever you're doing here is equivariant. So this entire side note basically tells us that one way to enforce equivariance in a message passing formulation is to ensure that messaging group invariance. And that is exactly what we follow while developing our architecture. Before we come to our model itself, we first talk about the interaction network, which serves as our benchmark. The interaction network is the current state of the art for the tracking problem. And it's an edge classifying network that produces a probability score for each edge in the head graph, which indicates whether the edge is true or false. Yeah, cool. So so now coming to the SO3 version of the equivalent, that is the SO3 equivariant version. This particular version takes only the node features as the inputs. As you can see, just the X's are inputs. EuclidNet consists of equivariant blocks, which are repeated multiple times to perform multiple message passing iterations. If you observe the leftmost image of the block, you'll see that we construct two scalars from the past vector, that is the inner products between the neighbors and the norms of the differences. These scalars are then used to construct the messages. So I think if you look at the first equation on the slide here, which indicates how the messages are created, you're using these inner products and these norms to construct the message. Now, after the message passing has been completed, these updated vectors are then pooled and they're passed through a decoding MLP to generate edge weights. Now, depending on which vector you pool, you can either pool the node features or you could pool the messages exchanged at the last message passing iteration. Now that would decide, so by choosing which vector to pool, you can choose to break equivariance at the final step. So if you choose a non-equivariant quantity and if that is passed to the decoding MLP, you've sort of break equivariance at that particular step. Now, the reason for including this particular feature was we wanted to study what effect this might have on the performance. So yeah, and this is a common feature across both flavors of EuclidNet. And finally, just to confirm if the network is actually invariant, you can look at the update step. It looks identical to what I discussed in the side note earlier. So any rotation matrix can sort of be factored out. So that tells us that this particular update follows the equivariance. Yeah, and then we have the SO2 equivariant version of EuclidNet. Now this is slightly different. Apart from node features, which are the X and Y coordinates now, not the Z coordinate, we have a group scalar, which is going to be the Z coordinate because that coordinate is unaffected by SO2 rotations. Now, these two quantities are passed as input to the GNN. Like in the previous slide, we construct these inner products and Euclidean norms. But this time we also construct another invariant, which is called E, which serves features, if you remember. But since they were non-equivariant, we couldn't use them. So that's what we're sort of replacing them by here. The messages are formed by using all of these scalar quantities. Yeah, and another difference in this particular version of EuclidNet is instead of using multiple blocks to perform the message passing iteration, weight sharing happens in recurrent neural networks. Iterations follow group equivariance, and like the previous model, there's also the choice to break equivariance at the final edge prediction step. One more tiny modification which we have is that in the first iteration, the scalars, which are the Z coordinates, are transformed via an MLP to an embedding space, just so everything is consistent dimensionally across all the steps we have. Cool. So that was the MLPs, the graph neural networks which we developed. But as one final test, we wanted to verify if the symmetries we baked into the network actually exist. To do this, we rotate our test data by applying a rotation matrix about the Z and the Y axes. And we pass the rotated inputs to our three models. This output is then compared to the rotated output, which is produced by the original unrotated data. If that's really confusing, I think this particular equation here should clear out what I mean. And so that error is basically what we plot as a function of the rotation angle. As you can see across these graphs, the interaction network sees a much larger deviation for both Y and Z rotations as compared to our EuclidNets. In fact, the errors observed for the EuclidNets are almost around the same order as the floating point precision we're working with. So that so it's safe to assume that the error is almost zero in this case. So this crude test which we have proves that the
Ameya, I think we lost your audio.
Actually based its inductive biases. Now at the final step, sorry, I thought someone said something.
Yeah, I did. You're all...
And interaction network really performs better here. If you want to see the exact numbers, yeah, although the SO3 EuclidNet has a smaller number of parameters than the ION, it's just unable to compete with the baseline. So this trend is seen for both 8 hidden channels and 16 hidden channels. The only advantage here is that EuclidNet has much better loss. It is possible that SO3 is too restrictive, and we might be able to get better performance with SO2. So that's what we do next. We benchmark the SO2 against the interaction network. Once again, both models have 8 hidden channels, and the EuclidNet has equivariant outputs. You'll see this time that the difference is really tiny. Although the EuclidNet being marginally better, the difference is actually really small. If you look at the numbers, that sort of puts it into perspective. The equivariant models don't seem to be as beneficial as we initially claimed they would be. The EuclidNet is still able to perform better with a fewer number of parameters, but there's still quite a bit of overlap between the confidence interval for both of these performances. And for a larger number of hidden channels, you see the same trend, but the interaction network seems to again dominate. Given these results, given the results people have observed with equivariant models and other areas of HEP, this sort of makes you wonder if the problem just isn't equivariant. If that's the case, any symmetric neural network which you use will always be too restrictive. So if there's any further work in this area, they would have to deal with this particular issue about unseen non-equivariances in the problem. And so to summarize, that really brings us to a close. We've developed an Euclidean equivariant GNN to the particle tracking problem, but early results indicate that it offers just a marginal improvement over the current state of the art because the results are still within one standard deviation of each other. And more future work is needed to concretely establish the possible reasons for this result. We think that this possibly the problem possibly contains non-equivariant aspects which cannot be learned by a trained model. Second, I think we presently have no known method to evaluate the quality of the learned equivariance, but there's some recent work which has the potential to let us know to the degree to which a network is invariant. And finally, there's some studies about soft equivariance which might be helpful. So yeah, there still needs to be some work done in this area before we can concretely establish that equivariant models are maybe not helpful this particular problem. In terms of deliverables, we have a poster coming up at ACAT about our work, and the code which was developed for this project is hosted on GitHub at that repository. So yeah, thanks a lot. It's been amazing to work on this project over the summer, and I've learned a lot. And special thanks to my mentors, Andrii Len and Savannah, for being so supportive and helpful for the project in helping me out with even the silliest questions or problems. Thanks a lot. Yeah, and I'm really sorry. I think I've gone over time, but yeah, thanks a lot.
Thank you, Ameya. Any questions?
Hi everyone, my name is Anni, and my mentors and I are from UC San Diego, and I'm a first-year undergraduate student majoring in physics. And today I would like to share my result of my summer project, the simulation of high-energy particle collisions using generative adversarial particle transformers. I would like to introduce my project first. So high energy particle collisions at the CERN LHC provide rich data, often containing jets for high energy physics research. And jets are sparse distributions of particles that have complex patterns, which we can use to identify rare particles, such as the Higgs bosons. But traditional simulations of such collisions consume a significant and increasingly unsustainable amount of computational resources. So we are exploring new machine learning methods to both improve and speed up these simulations. And among all these machine learning methods, graph neural networks have been found to be successful for a number of computational tasks in high energy physics, including generating realistic jets. My project is to implement a transformer-based generative adversarial network, and it's improved both the speed and fidelity of the generation. So what is the generative adversarial network? So GANs are a popular generative model in machine learning, and it includes a generator and discriminator, where the generator produces simulations and discriminator simultaneously tries to distinguish between real and generated simulations. And the generator and discriminator are trained together adversarially. So by improving the discriminator's ability to distinguish, we can simultaneously improve the generator's ability to produce more models closer to the real jets. And for the graph neural networks, so GNNs is a class of neural networks for processing data that best represented by graph data structures. And it can be directly applied to graphs and provide an easy way to do both node level, edge level, and graph level prediction tasks. And in our project, GNNs is used to be graphical representation, describes jets as nodes and edges, and combine them as a network. And in comparison to feed the graph of the jets directly to the GANs, the graph neural networks is less computational complexity and high efficiency with more information included. And yeah, since the high energy jets are sparse and have irregular shapes, it is better represented by graph neural networks. And the transformers are commonly used in language translation or conversational chatbots, or etc. And we noticed that there's an application of multi-head attentions, and the attention mechanism enables the transformers to have a long-time memory, focusing on all previous tokens that have been generated. And so it mainly takes advantage of the multi-head self-attention mechanism in the transformers. So many self-attention heads here composite a layer called multi-head attention layer, and each head can focus on a different subspace and modify the corresponding output sequence. So it will be more efficient. And I would like to introduce set transformers and our model GAPT. And set transformer, instead of normal transformer, it uses sets instead of sentence tokens as input. So it can be applied to jets. And compared with other methods for set structured data, set transformers can reduce computational complexity and have shown better performances. And we implemented set transformer into our previous GAN model, creating a generative adversarial particle transformer, which is GAPT. And there's 3 kinds of basic blocks in GAPT: MAB, SAB, and ISAB. MAB is multi-head attention block, a basic block used for attention-based set operations. So SAB and ISAB would all based on MAB. And SAB is set attention block, where we define this to be MAB XX, where X is the input n by D matrix, where n is the number of elements in set and D is the dimension. And ISAB is the induced set attention block. So we define an induced vector I, and it reduces the quadratic time and complexity for large sets. So the mechanism of ISAP is instead of taking care of X itself, there's the actual vector I, and I and X were firstly taken into MAB, and then the output and X again taken into MAB and create the output, and it reduces the time complexity to quadratic, from quadratic to linear. And we used Ernest as the data set to test the time and performance of this algorithm first, then we will move to JetNet dataset. So we have a basic sense of how transformers would be, and the next step would be implementing set transformers into our GAN. So this is the generator and this is the discriminator. So the generator takes the initial set, the number of elements n, and a noise z. And the initial set and the number n would be filtered. So the initial set would be filtered to a desired size using n. So the sample would be the same size as n. And Z and N would take into the multi-layer perceptron, and then the sample and noise will be put into the set transformer and generate the result. And the discriminator was very similar. So it takes the output of the generator and input data into set transformer. Then after the multi-layer perception, it would output the result. And this is our code for the generator in our gap. So firstly, initialize, and then there's some arguments, and the intermediate layers is like that, and the feed forward layer, and then finally use SAB to have the output. And similarly, this is our code for discriminator, and very similar, the initialization, the arguments, the intermediate layers, and the feed forward loop. And yeah, here we use the SABs and ISAP here in the intermediate layers. And this code is open to public for future use. And I put the link to the GitHub repository here. So we firstly run it on Ernest dataset, and the result of time looks like this. We run GAP using both ASAP and SAP to compare the time it takes, and it shows that if we increase the number of hits, which is the size of the set, to show how times will vary for these two blocks. And initially, the SAP takes shorter time than GAP using ISAP, but very soon, the ISAP is taking much shorter time than SAP does. And so it implies that ISAP, the induced SAT attention block, would reduce time very significantly. And we try to vary number of induced nodes for ASAP to explore the time further. And it shows that with M increases, the ISAP, the time taken by ASAP per epoch is gradually decreasing. But there are some outliers, and we guess that it is because of we're running different GPUs for each task, and they might have different performances. So this is the performance for gaps on Ernest. We vary number of induced nodes for ISAP and measure the fidelity and loss for the model. And it looks like that with M increasing, the fidelity also increases, but the loss of generator and discriminator remains roughly the same. And this picture is the best output for M equals 20. And we can see that it's pretty clear and can be considered as a good result. So then we move to JETS dataset. We run gap and message passing GAN on JETS dataset with number of hits equals 30 and number of ISAP nodes equals 10. So message passing GAN is a model developed by our groups previously. And I also put the link to paper here for further use. And we can see that for the same data set, the average time per output for gap is 28 seconds, but that for MP-GAN is nearly 3 minutes. And they have roughly the same loss for discriminator and generator. So this also implies that gap has a much better speed than other models for generating sets. And this is the result for the generation of JETs. And this one is the output of MP-GAN, and this one is the output of gap. So we can see that gap has a little like worse output, a less accuracy than MP-GAN does. So that was what we will do later for our work, is to adjust the code or modify the model to reach a better accuracy. But we can see that, yeah, it's pretty good in general. Yeah. And also these are the generation of particles using MP-GAN and gap. We can see that they are pretty similar in the accuracy, which is good because gap takes much shorter time than MP-GAN does. So conclusion, in our project, we implemented set transformers architecture in GANs to generate JETs, and we construct a promising model called gap. Based on Ernest dataset, gap reduces time it takes as well as maintains a good performance. And for our test on JETs dataset, gap also have a significantly faster speed than our previous models and has a reliable quality of the output. And some future steps would be first to finish tasks varying number of hits and ISAP number of nodes to explore deeper on JETs generation. And also we would adjust codes and tune the model to further improve the overall quality of the result. And we might combine gap with conditional GAN, which makes us be able to conditionally generate JETs with desired feature. And we're also going to refine the repo and add necessary instructions or comments in codes for public use in experimental collaborations. And thank you. And a special thanks to my mentors, Javier Duarte and Raghav Kansal, for helping me on this project. And thanks for IRIS-HEP to giving me this opportunity, and as well as the thanks for HSF training team for giving the training courses. Thank you.
Thank you, Annie. Nice talk. Javier, do you want to say a few words about Annie's work?
Sure, yeah. I was Annie's been working on this project, and it's gone in a couple different directions, and it's both kind of starting with looking at conditional GANs and now looking at this transformer architecture, which is really exciting and seems to be giving a lot of performance gains. So I think Annie is going to be kind of continuing to work on this, and I think it's a really interesting project, and Annie's done a great job. So yeah, thanks.
Right.
Thank you.
Thank you. Thank you again.
Right.
Before you get started, I'm gonna let's see. I see Henry and Mike are connected. Henry or Mike, you want to say anything about Elliott's work?
Rocky's here too.
Oh, Hi, Rocky. Sorry, I didn't see you there. Any one of you three want to say something? No?
Rocky, would you like to say something?
I guess you can better introduce him.
So Elliott was an undergraduate at Duke University when he started working on this PV Finder project. And during the summer of 2021, he was a IRIS-HEP fellow. And then this past summer, he worked as an IRIS-HEP fellow, continuing the work. And I'm very impressed by the results he achieved. I expect you will be as well.
All right. Thank you, Mike. Elliott, whenever you're ready.
All right, great. Thank you. Yeah, so my name is Elliott Kauffman. I'm going to be talking about PV Finder for Atlas, exploring a deep learning approach for primary vertex identification. Now, yeah, I initially I was working on applying the results for CMS and Atlas, but I have a limited time here today, and my progress with Atlas got a bit farther, so I think it would be more interesting for me to focus on that. And my mentors throughout this project were Rocky and Henry and Mike. So thank you to the three of them. They have been wonderful mentors. All right, so just an overview of the method. So the vertex finding approach is known as PV Finder, and it's a machine learning approach to primary vertex identification. So this method was initially developed for use in LHC-B data. They currently achieve efficiency values that exceed 98% for a pilot of 5.6 with a relatively low false positive rate. So the success that they've seen motivates the possibility of its extension to Atlas and CMS and other detectors. So this was sort of the motivation for my project. So the goal, as I've mentioned, use deep learning techniques to reconstruct primary vertices using detector tracks, so reconstructed particle tracks. And sort of an example of the output of this is seen on the right. So the input into the machine learning network is shown, or one of the inputs is shown in light green here on the right. And this is a kernel density estimator, or KDE. And it essentially represents track density as a function of Z, or along the beamline. And the idea is that peaks in this KDE are going to correlate with vertex locations. So we also use truth information from Monte Carlo data to calculate labels for use in the neural network. So that's seen in blue here in this example on the right, with the center being the truth vertex Z position and the width being proportional to the resolution. And then a convolutional neural network is used to predict the primary locations, the primary vertex locations. So this red distribution is trying to match the blue distribution, and we are looking to see if the centers of these two distributions are close together. So how do we do this? Well, starting from reconstructed tracks. So starting with reconstructed tracks, we calculate an object known as a Poca ellipsoid. So we used, for Atlas so far, we've been using truth-matched reconstructed tracks for training purposes. And we represent each one of these tracks as a three-dimensional ellipsoid. And a sort of a 2D cartoon of this is shown down below just for visualization as I explain this. The center of each ellipsoid is the track's point of closest approach, or Poca, to the beamline. And the size of each ellipsoid is proportional to the track's uncertainty. Therefore, a larger ellipsoid is going to correspond to a track that is not as well known. So smaller ellipsoids are going to contribute more to the kernel density estimator, which I'll explain more on the next slides. And it's basically the effect of using Poca ellipsoids is that tracks are only going to contribute significantly where they are closest to the beamline, and this is ideally closest to their parent vertex. So I developed this visualization so to see POCA ellipsoids a little bit more clearly. So these are two projections of these 3D ellipsoids onto the X, Z, and Y, Z planes. So as you can see, we have some larger ones and some smaller ones, and the larger ones are going to contribute less to the KDE. Therefore, but when we have these darker regions with multiple overlapping ellipsoids, they're going to have higher track density and more of a contribution. So, and the primary vertex is actually shown in blue here. So where you see a lot of overlap and a darker color in the ellipsoids, and you see that there is actually a primary vertex there. So how do we calculate the kernel density estimator for this? Now, this example I have a GIF of here on the right is actually from LHCb Monte Carlo, but it works the same in Atlas. So for each POCA ellipsoid, a Gaussian probability is calculated. And then for each bin along the z-axis, the probabilities of the contributing tracks are summed. So as you can see here on the top, the red line represents the bin that we are currently at, and then the bottom two plots here show the transverse plane at that point. And then the bars represent the track density that was calculated as the sum of all of the ellipsoid contributions. So for each Z-bin, we locate the point with the maximum track density. So that's the point that is shown in red that keeps jumping around here. And we record its height and position. Now this is first done with a coarse grid search as shown here, where we have 9 by 9, or it's a little bit more for Atlas, but we have a grid in which we find an initial point for the maximum, and then we use MinWe to find a more accurate position from that point. So the height becomes the KDE value for that bin, and then the position of the maximum, which we call Xmax and Ymax, are also used as features. Okay, so just a summary of the input features we use for the neural network. We have KDEA, which is shown in green here on the right, which is the sum of probability, the sum of track probability. We have KDEB, which is the sum of the track probability squared. And KDEB is essentially used to accentuate some features of KDE, the KDE that to aid in the training process. And then on the bottom here, we have Xmax, which is the X coordinate of the KDE value for each Z-bin, and then the Ymax, which is the Y coordinate of the KDE value for each Z-bin. So you can see that where there is not a primary vertex, Xmax and Ymax are far from the beamline, and then when there is a primary vertex, you have some like rapidly changing values that are close to the beamline. So this is the architecture that I considered for this study, is the UNet architecture, and it was initially developed for use in biomedical image segmentation. So this is an adaptation of that. Of course, instead of two-dimensional images, we have and layers of those, we have one-dimensional kernel density estimators and multiple of those. So it has parallel downsampling and upsampling steps, and these layers that are at the same depth are connected by skip connections that are performed via concatenation. So I'll go on to talk about results. So for training, I use a TT bar sample from Atlas Run 2 Monte Carlo, and I split it with 4,000 events for training and 10,000 events for validation. So this is just an example of PV Finder for Atlas working. So we have a true vertex here in blue, and we have a red vertex or a predicted vertex in red, and they closely match each other. And you can see how the neural network used this green input distribution, the KDE, to output the final results. So now actually talking about performance metrics. So I developed some methods to compare to Atlas's current standard algorithm for reconstructing primary vertices. And I should preface this by saying that PV Finder does not currently include assigning reconstructed tracks to its output predicted vertices at this moment. We want to do that in future studies, but that Atlas's standard performance metrics for their AMVF method rely on track assignment. So I develop a sort of non-standard method of comparing them. So I want to make it clear that I am not trying to make an extremely robust comparison at this point. I'm more trying to motivate that PV Finder is worth for further fine-tuning and investigation in the future because of how it currently compares to AMVF using this non-standard approach. So this is the first thing that I looked at. So it's the vertex resolution. So the resolution between pairs of nearby reconstructed vertices. So we see AMVF here in the red and PV Finder here in the blue. And these are counts from 10,000 events. So there are more primary vertices that are predicted closer together in PV Finder. So this would indicate a smaller resolution value for PV Finder, which is promising so far. We're currently trying to understand why that is. So we have this sigma 1 and sigma 2, where sigma 2 is 0.658, sigma 1 is 0.375, and those are used to determine our future or the upcoming performance metrics. So this is the non-standard classification scheme that I helped develop for to provide a comparison to the standard Atlas method, AMVF. So it involves coming up with 4 categories of predicted vertices: fake, clean, merged, and split. So this is my process. First, I iterate through the reconstructed vertex positions. I find the list of truth vertices with a Z position that is within sigma vertex of the reconstructed vertex C position, where the sigma vertex is taken directly from the values on this previous slide, sigma 1 and sigma 2, for PV Finder and AMVF respectively. And then if this list is empty, the reconstructed vertex is classified as fake. So if there are no nearby truth vertices, then it is a false positive. If the list has 1 entry, then that is a 1-to-1 assignment. That reconstructed vertex is classified as clean. If this list has more than 1 entry, so if it is sort of in between 2 truth vertices and sort of splitting contributions from both of them, that reconstructed vertex is classified as merged. And then finally, after we're done looking at all the reconstructed vertex positions, we iterate through the truth vertex assignments. So if a truth vertex has more than one assignment to a clean reconstructed vertex. So it's sort of the inverse of the merged case. Basically, a truth vertex is split between multiple predictions. Then all but the closest reconstructed vertex are reclassified as split. And this scheme was developed to be similar to the AMVF classification scheme, but it is obviously different because we don't associate the reconstructed tracks to the vertices yet. However, I did run this classification scheme on AMVF results so that we can at least show that comparison. So if you want to read how what their classification method is, I have a link here to their paper. So number of reconstructed vertices as a function of pileup. So this is just how many vertices are reconstructed from each category, and this is comparing PV Finder to AMVF. So PV Finder is on the left, AMVF is on the right. And then for reference, I have the pileup profile shown in the back here. So we have the majority of pileup from the sample is between 20 and 60 vertices per event. So you can sort of see that reflected in the error bars as well, as just for reference. And then this black line is just y equals x. It's just, that would be 100% reconstruction efficiency, which is not possible. And then we have our reconstruction acceptance shown in gray here. So not every vertex is going to be reconstructible, so that accounts for that. So we see on it's a bit difficult to see on this slide, but I'll show you in the next slide that AMVF seems to reconstruct more vertices overall, but at high pileup, PV Finder seems to predict more clean vertices. So that's a one-to-one association between a truth vertex and a predicted vertex, and AMVF has a higher false positive rate at high pileup. So this shows the ratio of the two. So the ratio of PV Finder to AMVF, and I wanted to include a horizontal line at 1 so we could see a little bit more clearly. But so as you can see, if we look at the total in black, this black ratio is just a little bit under 1 in every case. So AMVF does reconstruct more vertices overall. However, as you get to higher pileup, PV Finder so far seems to have more clean associations. So this is definitely worth looking into why this is, and it's promising for further studies and further development of PV Finder for the ATLAS experiment. And then finally, we looked at efficiency as a function of the number of associated truth matched reconstructed tracks. So efficiency is calculated as the number of clean matched truth vertices plus the number of merged. So all of the ones that are associated to reconstructed vertices, and that's divided by the total number of truth vertices. So we see this as a function of the number of truth matched reconstructed tracks on the bottom. So we see that they're basically past 20 years, so you have an efficiency of 1. But before that, we can see that AMVF currently outperforms PV Finder for low multiplicity tracks, low multiplicity vertices. So that is also a point that PV Finder can work to improve that in the future. But this is sort of just preliminary where we are at currently. And one current reason for this is while training for PV Finder, in ATLAS data, I'm currently requiring that each candidate truth vertex have at least 5 associated reconstructed tracks, truth match reconstructed tracks, in order to factor into the cost function. However, we can experiment with lowering this constraint and seeing if PV Finder can perform better in those regions in the future. So conclusions, as I mentioned, PV Finder appears to achieve better vertex-vertex resolution than AMVF, which is a good sign and worth further investigation. And AMVF performs better for PVs with lower track multiplicity. So that's an area in which PV Finder can try to improve through fine-tuning. And under this new classification scheme that was developed sort of as an intermediary before we do the track association to predicted vertices, PV Finder produces more clean reconstructed vertices at high pileup, while AMVF produces more total reconstructed vertices. So future studies, so not a lot of fine tuning for the neural network parameters was done yet. So adjusting those parameters, such as the depth of the network, how many layers, etc. And the input features can be tuned further. So the number of bins to use, the width of the bins, how we calculate the target, those can be fine-tuned in the future to try to increase the performance of PV Finder. We can also, as I mentioned, train on lower multiplicity PVs to try to increase performance. And we can use PV Finder output as seeds for assigning reconstructed tracks to reconstructed primary vertices. And this is a necessary thing to do because it's necessary for physics analysis and full kinematic reconstructed of the particles of interest. And it will also allow a better, more fair comparison to the current AMVF method. And because we can use their classification method. So, yeah. I would like to thank this long list of individuals for their wonderful contributions to this project. Rocky Balakarg, Lauren Tompkins, Mike Sokoloff, Simon Akar, Henry Schreiner, Michael Peters, Will Tepe, and Rita Sheehan. And I have my references here and with links in case anybody is interested. And thank you all for listening to my presentation, and thank you for this opportunity to work as an IRIS-HEP fellow. I'll take questions at this point.
Thank you, Elliott. Nice talk. Any questions for Elliott? No. All right. Well, that's all we have for today. Thank you to all the fellows for the presentations. Thank you for all the mentors for your work as well. That's all the presentations for this year, and we'll look forward to another crop next summer. So thanks.